first want to thank all the people who put together this conference for inviting me to speak here. Thank you so much. It's been great, and I really appreciate the time to come and take a day away from the VA and really stop and think about what is the work that we do here. I'm not going to talk much about the VA this morning, although pretty much everything we talk about here has implications for the veterans that we treat. I'm going to talk more about research that I've done along with Dr. Carrie Ressler, Dr. Tana Juranovic, Dr. Elizabeth Bender at the Grady Trauma Project in Atlanta, and a little more about, try to step back and take a bigger picture look at thinking about genetic and gene by environment influences on risk for stress-related and trauma-related psychopathology. So we will focus some on post-traumatic stress disorder, but also, as we learned yesterday, that post-traumatic stress disorder is just one of the many different outcomes of child maltreatment and other traumas that people experience over the course of the lifetime, and that many other disorders are also involved. All right. So did I hit the wrong button to make it move? Let's see. All right. Let's try it again. I'll just go the old-fashioned way. See if that works. No. What did you do? That's what I did a minute ago. Okay, I don't understand. Hopefully it will work better on the next slide. So a lot of what I'm going to start off by saying I think that we already know, but we'll just do a big picture foundation, which is that we all know that when we talk about trauma and stress-related psychiatric disorders, that some of the risk comes from exposure to stress and trauma and the environment the person is in, or even before the person is born. But some of the risk also is heritable and comes from the genetics with which they're born. And so we also know that, we know that genetics plays a role. We also know that exposure to stressful life events, particularly as we learned yesterday, early in the life, early in childhood and moving across adolescence, have a particularly strong impact in terms of risk for developing psychiatric and other behavioral and really physical health disorders across the course of the lifespan. But neither one of those factors alone will account for that variation. And so one of the next places we tend to look is to look at these two things together and to ask ourselves, how does the genetic code we're born with interact with our environment and the stressors and the other things we experience to contribute to risk? I mean, I think most of us who work at all with patients in the mental health field or even look at children that we raise, right, know that there's a combination of genetic and environmental factors at work with everything that goes on. But that has not been nearly as easy to study and research in psychiatry as it would seem like it is or should be, like many things. I think that one of the challenges has been over the last decade plus, there have been good many studies in psychiatry in particular looking at gene by environment interactions predicting outcomes. So starting with studies looking at exposure to stress and trauma predicting depression and other behavioral problems in children, and then moving through a number of areas, we've shown that different environmental stressors interact with specific genes or single nucleotide polymorphisms identified from the genome to predict risk for psychiatric outcomes. The challenge in this research has been that despite the fact that very often the genes that are selected the selections are driven theoretically based on what we know from animal research, based on what we know from other areas, when other groups try to replicate these same studies, they don't get replicated. Now this is, of course, we now know a science-wide problem and not a gene-by-environment problem, but it's a particular problem for gene-by-environment research, and that's led folks to question, what is the best way to figure this out? And I'm not going to come here with the answer to that question today, so we can stop that thought, but I'll talk with you some about our ideas. And so people have started looking at it, and you heard earlier talking about the methylome and looking at the entire process of methylation across the entire genome. And the same thing is true in psychiatric genetics. There's been a move towards looking across the entire gene, across the genome, to see if we can sort of in a theory-neutral way use statistics to point us in the direction of variations in genetics that increase risk for different psychiatric disorders. And that research has yielded some, but the reality is that at least so far in the area of PTSD, in the area of depression, and some of the other more stress-dependent diagnostic categories, although most of them are stress-dependent to some degree, including psychosis and bipolar disorder, which also we haven't really yet found a lot of hits in terms of looking across the genome either. And there's a lot of reasons for that. One of the biggest ones is that the N's that we're studying still need to get larger. Because in order to look across all of the genes and then correct for error, you need a very large sample. This is also actually true for looking at single gene polymorphisms in order to identify the effects that are likely very small, but people don't think about that as often. When we find them, we're often finding them in what we call underpowered samples. And so as a result, that sort of is maybe part of the problem with the instability of the findings. Another big challenge in GWAS, or genome-wide association studies, is that although people are working on it, for the most part, we haven't figured out a way to integrate environmental factors, because now you can imagine what a very large analysis is. It's one thing to try to figure out what are all the variations in the genome, and that's, we continue to get more and more refined methods with more and more variations available to us. But a whole other thing to try to figure out what are all the possible variations in the environment. That's a tremendous amount of, how would we begin to count that? And people have tried to do that, but that, I think, makes looking at the genes seem relatively constrained in terms of what you have to do. So why would we bother with this anyway? What's the point in trying to put all of this data together into a big old data stew and sort it out and see why? And the answer to that is, there's several answers to this, but I'll give you some of the best one. And the first one is that multiple studies, we've already said this, that environmental stressors are huge predictors of risk. So we need to have them in our analysis. We know it's there. We know it plays a fat role. We can't just simply say it's just too complicated. We won't look at it. We can, but that's probably not a good idea for finding good, robust, meaningful outcomes, clinically meaningful outcomes. Secondly, we know already from our non-human animal translational and even human research that environmental exposures are associated with biomarkers that are relevant. So you've seen that research presented multiple times yesterday, talking about not only the, psychiatric diagnosis or symptoms, which as was just pointed out has a lot of variability and challenges to it, but also continuing to seek biological markers, whether it's brain markers, whether it's cortisol level markers. And when we see those changes in biology, it points us in this direction. And then finally, we know that in some cases, there may be genetic effects that won't be present if we don't look at the environment. If we don't take the environment and other factors about the groups we're studying into account, we could have genetic effects right in front of our face and not be seeing them because they're masked by the environmental variations. And so the simple way to think about that is to think about having two very different environments that have, in theory, almost opposite effects on things. And you could go looking for a gene, but that data could get washed out to nothing, right, in between these two opposite environmental poles, and you wouldn't see that the gene is, in fact, playing a role in outcome if in half the cases the gene is conferring risk and the other half is conferring resilience dependent on the environment you're exposed to. And so I think we have to sort of say that's the other reason to look at it, is we think somewhere in there are combination effects. And I just put up here an example, one of those studies that's been done looking at genotype and social support influencing post-traumatic stress disorder and hurricane survivors, right? And so what they found was they didn't find a main effect of the genotype here, but they did find an interaction effect. And in just a minute, I'm going to talk a little more about another study, and we've talked already today or yesterday about FKBP5, and I'll talk a little more about it. But this is another gene where, for the most part, where we sometimes find main effects, we often find effects mostly when we take into account the impact of childhood maltreatment. And so we're going to talk a little bit about that. So in order to do that, I want to take just a minute and talk about the Grady Trauma Project. You've seen some of our data talked about earlier in this conversation, but I want to talk about it a little bit more in detail because understanding who the sample is and what their experiences are really helps us understand what is the data that we found, and that's sort of one of the major points that I will try to bring home at the end of my talk today. So first, you see all the names on this board, and this doesn't even begin to count all of the people that we work with day in and day out to gather this data at the Grady Trauma Project. We have colleagues now at McLean, Dr. Ressler, who's also at Emory, but mostly at McLean as he's moved to Boston. I'm still struggling with that, but that's all right. Dr. Jovanovic, Dr. Bender, Dr. Gillespie, Dr. Smith, and we can just run down the list of collaborators that we have. And not making it to this list, as discussed first thing this morning, are the hundreds and hundreds of people we've had over the last decade plus who are the ones who get up in the morning and go into an inner city hospital and sit in the primary care and obstetric gynecology clinics and walk up to people they don't know and ask them if they're willing to participate in research. And if they say yes, we say, great, we're going to need to ask you about 150 very personal questions, and we need you to spit into this tube for us, please. And then on top of that, finally, we want to thank the thousands of participants who have, for some reason, been willing to do that for us. Because I'm always awed that we can walk in and meet people we've never met before and explain to them what we're doing and have them willing to answer questions about childhood abuse, about other traumas, about their current psychological functioning, and then also give us spit all before they get to their doctor's appointment for today, right? And that's greatly appreciated. And so both the subjects and the research assistants. And then, of course, I would be remiss if I didn't list our funders here also, because without all of their support, we wouldn't have been able to do this research at all. So what are we doing? We're looking at civilian urban trauma. The big picture is understanding the genomic structure of PTSD. We have ongoing GWAS study. We have a sample of now probably more than 8,000. For the most part, it says here all traumatized. That probably depends on how you define trauma. It's somewhere between 85% and 95% traumatized, depending on whether you include witnessing, not witnessing, but highly trauma-exposed population. There's about 60% female, and 25 to 30% at some point in their lives have met criteria for post-traumatic stress disorder. And at any given time, if you walk into the waiting room of this urban hospital, the study actually started when we did a research study in the psychiatry clinic and found that a very large number of patients had PTSD, although only a very small percentage of those had PTSD in their charts. They often had psychosis and other things, but didn't have PTSD. Now, they may have had both disorders, but the PTSD was being systematically missed. So someone said to us, that the only reason you found that data is you're looking in the mental health clinic. And I didn't think that was so we went and started asking in the primary care clinic. And we found slightly lower, but not much lower rates of current, I'm not going to call it diagnosis because we're using a screening measure, but I would call it clinically significant symptoms of PTSD that are about 10 to 20% of the people, depending on how you want to slice the off-on button for what makes clinically significant. And those are rates that are comparable to returning veterans. Only these are civilians waiting for their primary care appointments, which tells you a lot about the environment. I won't spend a lot of time talking about it, but we've worked very closely with the Psychiatric Genomics Consortium, looking at PTSD, and we have a wide range of biological and behavioral data that goes along with our data. So what do we know about this population? I just told you they're highly traumatized, so I won't spend a lot of time on this, but somewhere between 20 and 30% have experienced each of these different types of trauma, including witnessing people attacked with weapons, physical assault, natural disaster. And the first time I saw the natural disaster data in here, I was a little confused as to why. It's also true of our house fire data. Why would we get such high reports of these? Why were these events so notable? I grew up in Atlanta. I wouldn't say that I'd, the same level. But when you don't have resources available to you, the impact of all of these things become greater. So in the neighborhoods our folks live in, when it rains a whole lot, their streets and their houses flood because the sewer system doesn't work as well in their neighborhoods. So every disaster, they're higher risk for house fires because they use ovens and other things to heat during the winter if they can't pay the heating bill. And so you become at higher risk. And so that is to say that all of these traumas occur in the context of other quote-unquote stressors. So it's not just neutral to the trauma, and this is where it gets complicated thinking about the environment. So many of them live in unsafe neighborhoods. This has been true since probably, subject 200, and the data hasn't changed much. In the last year, about 10% of our subjects have a close friend or family member, so someone that they know who's been murdered, and in their lifetime, 40% have. So it's a lot of exposure to things. Exposure, and this shouldn't be a surprise at this point, but it's an important point for thinking about gene by environment and research on trauma, which is that exposure to multiple types of trauma is the exception rather than the rule in our sample. I mean, it's the rule rather than the exception. I was wishful thinking there for a minute. Trauma isn't distributed randomly across the population. People who are at risk for one trauma are also at higher risk for other traumas. And this is a great example of what it looks like, with more often than not are people having 2 or 3 different types of trauma or 4 or more types of trauma. And when we say types, we actually mean types. We don't look at incidents. So you could have experienced more than one car accident or more than one assault, and it would still go in this data as one type of trauma. And this trauma usually begins more often than not in early life. So we have relatively more of our percentages who've experienced their first trauma in adolescence. Not surprisingly then, we'll also find that a large number of the people in our sample have been exposed to family violence and child abuse, including physical, sexual, and emotional abuse, as well as witnessing violence between caregivers. So we're looking at, for physical abuse, about 20% of our sample, up to as high as 27 to 30% of our sample for witnessing violence between caregivers. So this is, without a doubt, a very highly traumatized population, and that's important in understanding our data and thinking about how do we make sense out of it. So that's the picture of who are the subjects that we bring into this sample. And I haven't put the demographic data up here other than to say that, you know, it's 98% African American. It's almost all impoverished, with the vast majority living well below the poverty line. The hospital is a public hospital. The people are generally seeking because they don't have other resources for health care. All right, so that's the sample. Now we're going to take a minute, take a deep breath. I always deliver this data really fast about all of the horrible things and the murders and the traumas, and then I look out at my audience and people are sort of looking like deers in the headlight because I've presented this data a lot of times. And so take a minute and shift and sort of absorb the tremendous amount of trauma that our folks are experiencing. And now I want to talk a little bit more. So that's the sample. Now I'm going to move us back to the genes and talk about the FKBP5 variation. So we've talked about FKBP5, so I don't need to spend a lot of time here today discussing what it is, but it's a gene that plays a role in our stress response system. The way I explain it when I'm trying to explain it as simply as possible is to say we have the stress feedback loop that essentially serves as a thermostat for our response to stress. And of course, when we face actual genuine stressors, including the people that we're working with in this study who may face it on a day in, day out basis, we're supposed to have an upregulation of our stress response system. When we're no longer facing the stressors, it's supposed to downregulate. And FKBP5 is a gene through its influence that impacts this downregulation process. And we won't talk a lot more about that because that's been done already. What we do know now is that a number of studies looking at FKBP5 polymorphisms or differences in the FKBP5 gene have shown us that it interacts with childhood trauma, so not so much on its own, but taking into account data for childhood trauma exposure to predict risk for a wide range of psychiatric disorders, for PTSD, major depression, suicidality, aggression, and psychosis. And so we can ask a question about that finding, which is that a problem for your data, right? That it's not just PTSD, and I'm not particularly bothered by it in the sense that we've talked yesterday and we'll continue to talk more about early life stress, particularly maltreatment, as a risk factor for a broad range of psychiatric disorders. In fact, it would be sort of surprising to me if you found something that impacted an interaction with early maltreatment outcome and only impacted one psychiatric disorder, because the symptom overlap and the risk overlap profile is so great that it would be a little odd to see something that only impacted PTSD but didn't touch depression or didn't touch substance use, because there's so much overlap in those disorders and in the processes underlying them. Probably do better if we looked at, as some people have, the cluster of symptoms across the disorder and sort of sorted them out empirically and saw, you know, are there more differences in externalizing versus internalizing profiles than we will by looking simply at psychiatric diagnosis. So that particular finding hasn't bothered me. Here's an example of our paper on this where we looked at FKBP5 genotype predicting childhood abuse, interacting with childhood abuse. So this is a gene by environment interaction, and it's looking at adult PTSD symptoms. So we're looking at the PTSD symptoms here in adults, right? And then here is what we see in terms of the genotype here. And what you see is that this impact of the genotype really only comes in at this level of childhood trauma. So the way we've done it, we looked at the childhood, the CTQ, and we divided up by no childhood abuse, one type of childhood abuse, or two or more types of childhood abuse. And only at these higher levels of abuse do we see an impact whereby the gene, it's almost like a dose response with the alleles and the outcome. Some variants of this genotype appear to be protective in the face of higher stress, or at least, if not protective, mitigate risk to some degree, and others tend to be associated with a much worse outcome. And this was talked about earlier today, but this is our research group also talking about, so how does that happen? What's going on with that? And we talked already, so the stage was set very nicely for me today, which is to say that what we're thinking at this point is that the childhood environment interacts with the genotype to change the methylation and the other epigenetic processes that didn't change how the gene expresses itself. And that has an impact on the stress response system. So here is some data that was nicely laid out showing that FKBP5 genotype, so what variant of this gene you have, interacts with your childhood trauma exposure to alter the methylation of FKBP5 in a way that ultimately alters the expression of this same gene and then affects your stress response system. So this is how, we think that it is working, at least as we've learned so far. And then we've also continued to look at, across our group, other factors. And so this is a study that's done in our group looking at hippocampal activation and FKBP5 risk allele carriers. And we see differences in brain activation during processes of threat, congruent versus threat incongruent phases, depending on whether you have a risk allele or don't have a risk allele for this gene. So we won't get into the depths of all of this because that could take me the rest of the day, but we'll say is that this is a continued, demonstration of the different ways. And then this is not our study, but another study showing that FKBP5, someone asked yesterday, had emotional neglect. And we'll talk in a minute about, is it neglect, is it abuse? What do we make, as we talked yesterday about this 2 or more types of abuse, how do we understand what that data means? But this shows that it also predicts individual differences in amygdala reactivity. So what we've seen is that multiple SNPs in FKBP5, in this case, show a significant interaction with emotional neglect on, this is right dorsal amygdala activity in the case of this study. And it continues to sort of demonstrate for us that we're seeing a biological outcome of this genotype in interaction with childhood environment. So what's the conclusion on FKBP5? So multiple variants of this gene interact with severity of childhood abuse to predict adult risk and resilience for trauma and stress-related psychiatric disorders. We think this mediates risk by altering glucocorticoid sensitivity. It's associated with differential hippocampal and amygdala function, interacts with childhood trauma to alter methylation and expression. And so all of this together, I think, pretty robustly suggests a case for gene by environment interaction in predicting response, right? So here I think we see a case where different areas, although this is not a gene that has demonstrated itself in genome-wide studies of post-traumatic stress disorder, why wouldn't it have done that if it's playing the role? The answer is it doesn't demonstrate itself until you look at childhood trauma levels. And so unless you can put those 2 pieces of information together, you're not going to see its effect. All right, so this is sort of the third component of my talk. I probably needed slides to sort of give us space in between to allow us to shift along with it. What time is it? So I can pace myself. Okay. So now I'm going to spend a little bit more time talking about this concept of the environment, right? When we talk about gene by environment interactions, right? And what do we mean when we say the environment, especially in research on childhood maltreatment? And we started having this discussion yesterday, which is to think about types of childhood trauma and types of adult trauma and risk. And this is just a very simple slide, but I think it's telling, which is shows in our population that number of trauma types in both adulthood, so this is in our sample, the same thing we saw earlier, which is the groups with no trauma, 1 trauma, 2 or 3 traumas, or 4 or more traumas, and then this is your PTSD symptom level going across up here. So there's a clear dose-response relationship, which again, this is cross-sectional. We do have a, now looking at children and their mothers over time study, but for now, this is one of the things we talked about yesterday, is one of the ways you look at cross-sectional data for evidence, right, of causal effect, although it's certainly not proof. We see this incredibly robust dose-response relationship, just as we do with childhood abuse. And if I put up here, but I haven't, this interaction, what we see is that childhood trauma interacts with adult trauma exposure to spike the risk even more. Those 2 things go together. Now, given what we talked about yesterday, I want to take just a minute and talk about this thing, because we see this finding in different ways throughout our research at Grady Trauma and other areas of research, which is that if you look in the CTQ, which is a pretty commonly used measure in genetics research of childhood abuse, it's not probably as good as the more comprehensive interviews like we saw discussed yesterday, where you have more time to sit and map out every trauma, but it's fairly robust, and in fact, I've done that kind of interviewing, and it seems to correlate with it fairly well, and it's used in a lot of gene by environment studies, so that helps us at least hold some aspect of the research the same. It has the same limitations of all self-report measures. But we find that when people start reporting 2 or more types of abuse, and I've looked at this different ways, it doesn't, depending on your outcome variable and how you look at it, it's not so much which type of abuse, it's whether or not you have 2 or more types. And what does that mean, right? Does it really not matter whether you had sexual and physical or physical and emotional? Probably it does matter, we're just not smart enough to measure it, right? Because we saw yesterday very specific brain region outcome effects of different types of trauma. But I think more importantly, what this tells us is, what does it tell you about your overall family environment that you're raised in if you're reporting 2 or more types of childhood abuse? So maybe you had someone come through the household who was physically abusive, but everyone else in the house